High-affinity immunoglobulin G and immunoglobulin A antibodies can neutralize bacterial toxins. Many bacteria cause disease by secreting proteins called toxins, which damage or disrupt the function of the host cells. To have an effect, a toxin must interact specifically with a molecule that serves as a receptor on the surface of the target cell. In many toxins, the receptor binding domain is on one polypeptide chain, whereas the toxic function is carried by a second chain. Antibodies that bind to the receptor binding site on the toxin molecule can prevent the toxin from binding to the cell and thus protect the cell from attack. Antibodies that act in this way to neutralize toxins are referred to as neutralizing antibodies. Most toxins are active at nanomolar concentrations. A single molecule of diphtheria toxin can kill a cell. To neutralize toxins, therefore, antibodies must be able to diffuse into the tissues and bind the toxin rapidly and with high affinity. The ability of immunoglobulin G antibodies to diffuse easily throughout the extracellular fluid and their high affinity make these the principal neutralizing antibodies for toxins found in tissues. Immunoglobulin A antibodies neutralize toxins at the mucosal surfaces of the body. Diphtheria and tetanus toxoids are two bacterial toxins in which the toxic and receptor binding functions are on separate protein chains. It is therefore possible to immunize individuals, usually as infants, with modified toxin molecules in which the toxic chain has been denatured. These modified toxins, called toxoids, lack toxic activity but retain the receptor binding site. Thus, immunization with a toxoid induces neutralizing antibodies that protect against the native toxin. With some insect or animal venoms that are so toxic that a single exposure can cause severe tissue damage or death, the adaptive immune response is too slow to be protective. Exposure to these venoms is a rare event, and protective vaccines have not yet been developed for use in humans. Instead, neutralizing antibodies are generated by immunizing other species, such as horses, with insect and snake venoms to produce anti-venom antibodies, anti-venins, for use in protecting humans. Transfer of antibodies in this way is known as passive immunization. And that's high-affinity immunoglobulin G and immunoglobulin A antibodies can neutralize bacterial toxins. Have a great day.